some basic things that I have to disclose and disclaim before every webinar as people are gradually joining in. So welcome everyone who's already here. My name is Danish Bhatti. I'm the chair of APPNA Merit for 2021, the Medical Education, Research, Information and Technology Transfer Committee for APPNA, tasked or mandated with health education and research innovation in Pakistan. I am a movement disorders neurologist and associate professor at University of Nebraska Medical Center, and I'll be moderating the session today along with my co-chairs within the APPNA committee who have joined me. So remember, this is an ACCME approved CME activity. We encourage all the speakers to disclose their conflicts of interest. This is done as a learning opportunity only to enhance our understanding of the current field of research. And that's why we often invite industry leaders and various organizational leaders to present their work in this context without intentionally or unintentionally endorsing any activity. But we, of course, think of this as a networking opportunity to come up with projects and collaboration. So use it for that. Please introduce yourself in the chat box. Please leave your email if you want to be reached out. Many of the speakers also share their contacts and you can reach out to them and use this opportunity to network. We also invite anyone who wants to recommend themselves as panelists or someone else they know and speaker, please reach out to us at appnamerit@gmail.com. The CMEs will be issued once a quarter, not for each individual attendance, but we count all the attendance and we'll make sure that you get the appropriate CME certificate for all the attendance. And I also want to thank the APPNA RISA committee, the Committee on Research, Education, and Scientific Affairs, for graciously co-sponsoring this CME and covering all the costs of CME so the program can be offered for free. And we are also thankful to our partners in if I say it right, CPRD, Canadian Pakistani Research Development Forum. And Dr. Syed is representing it and is our collaborative partner for these webinar series. We will take some time to have some panel discussion afterwards and some question answers. You can put your questions in Q&A box. You can also add comments or questions in the chat box if needed, but we prefer using Q&A box if possible so that we don't lose your question. And if we have time, we will allow microphone to some of the audience who want to make a comment or ask a question personally if we are not running too short of time or not too much over time. With that, let me start with inviting our speaker for today. It's my great pleasure and honor to invite a true researcher in the field of neurosciences, Professor Dr. Naweed Syed. Professor Syed is a chief scientist at Creative Destruction Lab and Peak Scholar for Cumming School of Medicine, University of Calgary. Dr. Syed his postdoctoral program director at the Office of the Vice President Research from 2012 to 2016, and special advisor to the Vice President for Research. He has served as the research director of Hotchkiss Brain Institute and the department head for cell biology and anatomy for over 10 years, and has directed research at Alberta Children's Hospital Research Institute for 5 years. It's a great honor to have Dr. Syed among us for his excellent breakthrough research in brain chip interface, a truly remarkable pioneering work. And I'm really interested in hearing his work and future directions, and maybe we can have some discussion on how Pakistan can leapfrog into the front of research by jumping this new technology research without having to go through a buildup of old school clinical trials, which are much more resource intense and much more slower. So with that, I invite Professor Syed to make any more additional comments and share his screen if he would like. Please unmute yourself, Professor Syed.
Can you see my screen?
Yes.
So, Bismillah ar-Rahman ar-Rahim, Assalamu alaikum to everyone. It is an absolute really pleasure, privilege, and honor to be part of this really elite group. And I really commend the activities of the Research, Education, and Scientific Affairs Committee. And I'm really grateful to Dr. Danish Bhatti for his kind introduction, and also Dr. Mustafa Saad Siddiqui and Rashid Ahmed Bhai for being here, and all the other audience. I think, first of all, I'd like to give a disclaimer, a conflict of interest, that I really have no interest of any kind in any of these activities that I will talk about or present in my talk. I do have affiliations with some neuro companies, but none of those are really involved in the activities that I will present today. So I would like to really thank all the audience for being here. And since it's maybe the end of Ramadan, we are fasting, and I think our colleagues in Pakistan, I believe, have just finished their Taraweehs, and they are also exhausted as well. So what I will do is, before I run out of energy, I will really like to give a take-home message for everyone in my first couple of slides. So this is where I am from. Calgary is located in the foothills of Canadian Rockies. It's a beautiful city. We have spring really coming up, and not quite yet. Even living in this place, you wouldn't believe that it is snowing outside right now. So whereas people may be cooking in Pakistan and other places, but we have snow as we speak right now. So really the point of showing this slide is that even in countries such as Canada, which are really rich, resourceful countries, and they have tremendous opportunities, tremendous interest in research, innovation, technology development, commercialization, entrepreneurial activities, we also have challenges as well. And the challenges are of different nature than what people will face in other countries and other places. Now, but we don't just sit when we see our cars buried under snow, but we actually think about how we can solve these issues and get ourselves out of this parking lot. And for this, we build these interesting roads cutting the snow, and then try to get to another place where we really need to be. So I think the take-home message is that yes, there are challenges, and the challenges for third world countries are actually going to get worse for post-COVID, whereby that in Pakistan we are struggling with vaccine productions, ventilators, all the other things, and each and every country is having its own challenges. So it is time for Pakistan especially to really think and reinvent its way out of these troubles. And for this, I think that yes, there are challenges. Money is an issue. And as Jeff said, some times ago, the frugality drives innovation, just like other constraints do. Now, one of the only ways to get out of a tight box is to invent our way out of it. So I think this is really important, and this is a critical time that we can do a great deal for Pakistan and to help them really move to the front of the research frontiers. So if we don't push ourselves out of our comfort zones, then many of the disruptive technologies that are in the works in North America, in Europe, and other countries, they will really throw our local doctors and healthcare providers in Pakistan and in other developing countries out of business. And I also like to really give a take-home message that investment in knowledge always pays the best interest, as Benjamin Franklin once said. And not investing and adapting modern medical technologies will be devastating for developing countries such as Pakistan. So I think this is my take-home message, just in case I thought that I would run out of energy. Now to my own work. Now, I am a neuroscientist by training, and I'm really interested, have been interested in fundamental mechanisms as to how brain functions and what are the basic cellular molecular mechanisms that allow brain to generate these beautiful activities that we see. And when I was a department head, I was once called to do an autopsy on one of the young girls who died at our children's hospital. This young girl was perfectly okay. 2 days before her death, she developed some flu-like symptoms. Family didn't really care too much. They gave her a Tylenol. She went to sleep, but her situation got worse. She started to have seizures. She started to have other complications, respiratory problems. They rushed her to our children's hospital. She was taken into emergency, and they tried to do everything to this young girl. But in our intensive care, within 48 hours, she passed away. And it was really a shock because she was perfectly healthy before that. So the family and the doctors really wanted to see as to what might have gone wrong with her. So her brain was sitting in front of our table, and we realized at that moment, when I realized that I was looking at a piece of tissue, and that is just, maybe 2 pounds or so. And that was just a piece of tissue, which is the biological tissues. But I started to wonder about her sentiments and her feelings, that what made her smile. She was a very bubbly young girl. What made her love her family? Affection, kindness, and her ability to really think about future, her ability to solve problems, her feelings that she gets upset with her friends and then gets happy, smiling, upset, crying. All of those aspects, the feelings and beliefs, they were really not observable or measurable by looking at this piece of tissue. And that really daunted to me as to really, if we are ever to figure out how brain functions, the brain that has tens of billions of brain cells, more than the Milky Way, and these brain cells are then interconnected with trillions of synaptic connections, and even more complex really is the interplay between these brain cells. So if we are ever to figure out how this squishy stuff we call brain functions, we really have to tap into the information processing capabilities that is confined within this piece of tissue. And the other important thing that, that occurred to us, that after trauma, injury, Parkinson's, Alzheimer's, the natural replacement of damaged brain tissue doesn't take place. So how do you repair it? How do you fix that brain tissue when there is no other opportunities? Even for psychiatric disorders, we manage them, we don't treat them. And many other neurological symptoms are only manageable. And we can't even use stem cell therapy to repair brain because those stem cells turn into tumors. And even if we manage to produce neurons, these stem cells produce neurons are younger. They still have to reach out to other partners which are located at some distance, but the actual pathways that allow these brain cells to be guided towards target cells are no longer there. So the first really question that we always ask is that, how does our brain function and how can it be repaired when damaged? We also ask the question that, can this brain be connected with machines? Which is really an interesting question, and that will be the second part of my talk, as to how we can connect brain, which is very similar to computers, to connect it with computers so we could have a two-way dialogue between a machine and a human brain. And maybe that approach could be used to really regain or replace the damaged brain tissue. Science fiction, it may sound, but I will really walk you through as to how far we have come and where this research is leading. I will not talk about the major aspects of my research in using various different animal models and different approaches and technologies, but I will focus primarily on brain-machine interfacing component in this talk. But our research program is really focused as determining these neurons, or we call the brain cells, the fundamental unit of nervous system. How do they connect with each other through synaptic connections? And how do these synaptic connections give rise to neuronal circuits, neuronal networks, which then elicit a particular behavior? We also know that these neuronal circuits in human brain are not hardwired. They exhibit a high degree of plasticity, meaning that they change all the time. And every time we learn something, there are new synaptic connections formed, and these synaptic connections form the basis for learning, memory, and other aspects of brain functions, and to elicit really the behavior as well. Then there are these glial cells. The glial cells are no longer just the support units, but they are active partners in this tripartite relationship between different brain cells. And many of these neuronal circuits are really determined genetically. In humans, maybe up to 25%. But all the other aspects of brain wiring depends upon epigenetic. And these are the environmental factors that will help shape the final connectivity patterns of brain, which then elicits or modulate or control specific behaviors. We know that the human brain is the fastest growing organ in human body, and genetic perturbations or environmental perturbations in utero can render this growth susceptible to neurodevelopmental diseases. And this is one of the ways we find out how brain functions or how brain dysfunctions. So most of our information that we currently have about the functioning of human brain has come from when things go awry, when things do not work properly, neurodevelopmental disorders, we have autism spectrum, and many other aspects of it that we know or we understand from neurodevelopmental processes going wrong. We also know that the development or early childhood, neglect, trauma, and also abuse will render these networks of brain cells compromised for life. And here is a brain scan of a 3-year-old child, 2 of them, and the normal brain. You can see how extensive the size of the brain is and how well developed the brain is. And then the child on the right-hand side, you would find has gone through extreme neglect, abuse, malnourishment, and as a result, you could see the differences in the developmental patterns. And so this is how we find out how nurture, how epigenetic really plays an important role in defining the final patterns of connectivity. But the important thing also to note is that at early stages, if we can find, we step into these processes, there are ways to reverse some of these changes and will allow the brain to regain most of its functions through love, compassion, kindness, and providing these children with an enriched environment through which to learn. We also learn about brain functions indirectly and in the neurodegenerative diseases when the neuronal circuits just fall apart. And we know that the brain generally seeks a purpose through interconnectedness. So brain cells that fire together, they will wire together. But if this interconnectedness is missing and its absence, the neurons start to commit suicide. And we see this in psychiatric disorders, as well as we see in neurodegenerative diseases, such as Alzheimer's and Parkinson's and many other diseases. So we really don't have the means to repair a brain if this wiring goes haywire. And that is one of the biggest challenges in the field of neurosciences, neurology, and neurosciences, neurosurgery. In our lab, we find out the different functions of the brain, again, indirectly. So I have shown you that we know brain functions, which particular network controls which particular behavior through neurodevelopmental processes going haywire. We understand about neurodegenerative diseases as to how brain function may be perturbed. And then we use some techniques. For example, in our lab, we will use optogenetics, where we use channel rhodopsin. We will express these specific channel rhodopsin proteins into specific sets of brain neurons, and we tag them with different components. And then what we do is we shine light to activate only those neurons or inactivate those neurons, and then try to figure out how brain networks control a particular behavior, or if they do or do not participate, whether they are sufficient, whether they are necessary, or whether they are appropriate. The other approach we use in our lab is that we can also use gene knockout. So we can use very specific transcripts and we can tag particular subsets of neurons. When we inject these transcripts inside, we can knock out certain very specific brain cells in specific regions of the brain. And then we ask the question, what happens to animal in terms of their learning, memory, and other responses? So from optogenetics, where you have channel rhodopsin proteins expressed in the brain, to gene knockout, we begin to understand some parts of the brain function as to how brain may control these behaviors. But these are most of the time indirect, whereby you are actually using indirect means to be able to decipher function of particular subsets of neurons in any given behavior. We know also that the genetic perturbation and environmental factors, they play a very important role. During development, brain cells that fire together, they fire together. If you prevent that firing, we know that the brain circuit will not wire proper. So if you have a kitty cat, you put a patch over one of its eyes just for a couple of days during critical period in brain development, this kitty cat will now be blind for the rest of its life, because those brain cells that are reaching from eye to the visual cortex are not connecting or not firing together. So this firing together, wiring together is really important. If brain cells are deprived of any sensory input, some parts of the brain circuits will not develop. And this is another way that we find out how networks of brain cells, or which particular network of brain cell underlies which given behavior. And we also know that if neurons, they fire apart, they will fall apart. And neurons that fire out of sync, they will fail to link. So these are developmental and also adult related synaptic plasticity that really forms the basis for brain function. So this is really an overview of as to how we have come to know about various neuronal networks in different regions of the brain as to how they control specific behaviors, or they may or may not. But this is, again, indirect means or way of really deciphering the functions of brain networks vis-a-vis any particular behavior. So one of the important questions that we ask all the time is, how do brain cells really communicate? We are seeing the activity patterns are important, but how do they communicate? So for the sake of audience and simplicity, we know that at the synaptic terminal where two neurons that come together, we have really these synaptic connections between neurons that will allow the release of transmitters. So on one side, you have electrical impulses that are coming in. And on the other hand, you really have chemical synapses that are formed. So when you're looking at a typical chemical synapse between two neurons, they're surrounded by their extracellular milieu, which have various different ions, calcium, sodium, potassium. And at the terminal, you have these specific calcium channels or ion channels that are clusters at the nerve terminal. And when an impulse arrives at the presynaptic terminal, the voltage-gated calcium channels will open. They allow these calcium ions to come and mobilize vesicles, transmitter vesicles, serotonin, dopamine, to be really tagged at the synaptic site, where they are released from the presynaptic terminal. They bind to the postsynaptic receptors and open again, either ligand-gated or voltage-gated ion channels. So the impulse, which was electrical, becomes chemical, and then chemical to electrical again. So in this respect, the actual circuit behaves very much like an electrical circuit. And so we find out how brain cells really interact and communicate by culturing these neurons. Of course, you cannot do these experiments in the intact brain. We can culture these neurons in vitro in cell culture. We can use either intracellular recordings or patch clamp recordings, and we can then trigger action potentials in presynaptic neurons. And as you fire these single spikes, you can see the postsynaptic cell is exhibiting a postsynaptic potential, which is really a chemical now synapse between these two cells. But the interesting thing to note really is that when you fire a burst of spikes, you get a much bigger response. But if you wait for even hour, 2 hours, 5 hours, and do not allow this neurons to fire an action potential, what you see is a single spike will produce much bigger response. And we call it synaptic plasticity. And this synaptic plasticity, short and long term, forms the basis of all working memory and learning and memory, because the synapse will be primed and potentiated. So this is really at the 2 cell levels, but this wouldn't tell me how 2 neurons or larger networks of neurons control a particular behavior or functions. So we then grow these large networks of brain cells, and either using calcium imaging, voltage sensitive imaging, we try to decipher how these brain cells interact with each other, but we are still limited in terms of as to how many brain cells we can record with these conventional intracellular recording techniques at a resolution of single synapses or synaptic contact. So that really was the grand challenge that we took upon ourselves, that if we are to figure out how brain controls these large networks, together, working together, control a particular behavior, we have to develop technology and techniques that allow us to record from larger networks of neurons. So this is where the brain chip concept really comes in place. But to be able to really connect or understand, we first needed to take the information that I provided you earlier, specific subsets of neurons, either from the cortex or from the hippocampus, and we needed to really use imaging to figure out how these networks function in brain. And then we asked the question, can this now be recapitulated in vitro? And then could these networks be interfaced with semiconductor chips or an artificial device? And what you see in the clinical settings is that right now, there are usually always one-way communication between brain cells and any semiconductor or a foreign electric device. So for example, people who have Parkinson's, they don't respond to drug, the surgeon will decide to plant a deep brain stimulation electrodes, surgically implanted in the areas where the dopamine cells are dysfunctional. And what they do is that the electrodes can stimulate those brain cells. The brain cells will be activated. As a result, you alleviate some of the symptoms, and you can activate and deactivate these electrodes at will whenever you need it to perform certain tasks. And they really work quite very well in most instances. But the situation here is that the electrodes continue to stimulate those neurons, and it ends up often causing excitotoxicity, and also ends up causing really excitatory glutamate-induced damage to brain cells. These brain cells are screaming, stop, stop, stop. But there is really no means to stop the stimulation because the electrode has only one way, one direction. In the instances of epilepsy, when you plant these electrodes in the to monitor epileptic seizures on the scalp of the brain, the surface electrodes, whenever a seizure foci will discharge, these electrodes pick up the signal, and then the surgeon could decipher as to where the seizures are coming from. But these are also one-way talk, one-way link from neuron to a transistor or a electronic device. What we really wanted to do was that instead of using a stimulator, a conventional stimulator, we wanted to use a semiconductor capacitor that could stimulate a brain cell. And if this brain cell is now discharged and fires and it's connected synaptically with its partner on the other side, when this neuron discharge, you have a transistor underneath that picks up a signal. So you complete a loop whereby a electronic device can talk to brain cells and brain cells can talk back to it. So the idea really is that if because of stroke, trauma, or injury, these two brain cells had lost their contact, we can really bypass by using electronics to be able to reach out to this neuron and then really get it to perform function. I think this part of neuronal dynamics and digital computation is maybe 20, 30 years from now, and we are really not touching. So the way it really works in instances of our original chip was, the idea was that these brain cells, chips are connected to computer, and underneath the neurons, we have a capacitor. We generate capacitive current as if one neuron is activating another neuron. And the neuron will fire as if it is being depolarized by another neuron. So once the capacitor activates, the cell fires an action potential, it triggers and activates the postsynaptic neuron, and the transistor underneath picks up a signal. So you actually complete a loop whereby an electronic device can talk to brain cells and brain cells can talk back. That was really the prerequisite for us for any brain controlled prosthetic devices or brain machine interfacing. So when we cultured these neurons on a semiconductor chip, the biggest challenge was to develop a substrate that would allow us to be able to make sure that these neurons are happy. They are growing as if they were growing in the intact brain. There were no really issues in terms of toxicity. So we had to make these chips biocompatible, and that was really involved a lot of chemical engineering to come to that stage. And we were really delighted to see these brain cells growing beautifully. But then the next part was really to keep these brain cells alive for quite some time. And we developed these incubator systems, which have CO2, PCO, oxygen, humidity, temperature control, to make sure that we could study these brain cells outside for a very, very long time period. And this allowed us to be able to really grow these brain cells as we deemed appropriate by creating these patterns on the chip, whereby brain cells will only grow between, on these patterns, and underneath each of these patterns is either a transistor or a capacitor that can pick up the activities. And we can map these neuronal networks and the activities at the level of individual neurons for a very, very long time period. And this was really a major issue for us to be able to crack that code whereby we could monitor activities of these brain cells. So of course, these brain cells that we dissociate are culture, they may not connect the same way as they connect in the intact brain. So because they are most of the time really artificially dissociated, we take brain, we dissociate them, and two cells that we find are connected may not be connected in the brain the same way. So the next task that we wanted to do was to see if we could monitor naturally designed neuronal circuits on our semiconductor chip. So here we develop 3D electrodes. They penetrate and they are contacting individual neurons. And then we could chemically induce seizures, and the activity or spontaneous activity could be really picked up by these electrodes at a very high resolution. And so this is really in the intact brain, but the question remained that we are only monitoring the activities of those neurons that have established contact with our electrodes, but brain is three-dimensional. What about brain cells or neurons that are sitting in layers? So for this, we developed another technique called photoconductive stimulation, where we grow these brain cells on silicon wafer. We label them with fluorescent dyes, which is voltage sensitive dyes. So when a brain cell fires, it will be activated, and then the voltage will change the actual properties of either calcium indicator or voltage sensitive dyes. So when we fire a laser through the microscope, it will hit the wafer. The wafer changes the properties of the neurons. The neurons are depolarized, and when they depolarize, they fire. And when they fire, we can actually monitor activities of large of these networks of neurons, millions of them at a resolution of single cells. So this was really a major aspect for us, that if we are to understand how brain functions, we not only need to record from those neurons, hundreds of thousands of neurons that are in immediately interface with semiconductor chip, but also those neurons that sit in different layers. So if you have this power put together, really, where does this lead? And then we are doing these experiments in collaboration with Tsinghua University in China. The idea is that in this particular case, for example, the older experiments that were done in the United States, you could see that the monkey's visual cortex will make contact with the palate. And this visual cortex, when it fires, it will activate the sensory motor cortex. And when the sensory motor cortex discharges, if you plant a chip in that area, that chip, because you have designed it, you can couple it with a prosthetic limb which you have designed. So making this process really, now our idea is to be able to plant these chips subcortically, so we don't interfere or perturb or affect those neuronal networks in the brain that are, controlling other functions. So we also know, as that, Andy Clark said, that we humans are naturally born cyborgs. So brain plasticity allows us to attach ourselves to machines and computers and electronic tools very, very naturally. We are connected to our cell phone is connected to a server is connected to our main tower, which is connected to satellite. So humans have the ability to naturally connect, and our brain reconfigures itself in response to an input from a simple tool that we use really routinely. So, and it is a far more open system than we had or ever imagined. And it also survives in an ever-changing environment. So if our brain is altered or adapts to a computer, an electronic device, it doesn't mean that it will change in a sense that it will not survive. It actually perfectly survives, and it reconfigures itself very nicely. So here's another interesting experiment whereby you can use really think about brain-controlled prosthetic devices where people tend to have phantom limb. And often the time we try to eliminate those feelings from the brain, that the phantom limb or the phantom pain is really not good. But I think in many instances, those types of aspects in the brain could be tapped in for some really good and very cool approaches to be able to connect not just to a machine, but any other object that your brain gets convinced that it belongs to it. So in this particular experiment, you will see that this individual is asked to focus on a rubber hand, which is sitting in front, and then his own hand, which is hidden from him. He's asked to focus on hand, and as his rubber hand that he's focusing on is stroked, his real finger is also stroked at the same time. And watch what happens.
Practice. Here you can see the fake hand focusing on it. There's the real hand not focusing on it, simultaneous stroking. And there are various ways you can test it.
Let me show you again.
Practice. Here you can see the fake hand focusing on it. There's the real hand not focusing on it, simultaneous.
Oops, sorry. So you got the idea that really even brain is empowered in a sense that it could imagine a prosthetic limb or artificial limb. So by just thinking about it, you could link the brain with a particular rubber hand, let alone an electronic device. So brain machine interfacing is, if I really now think about reaching out to grab my remote control, the parts of the areas in my sensory motor cortex that discharge when I do this action, they will be activated when I only imagine about this. So I think even in our dreams, when we dream about punching someone, the same part of our brain is activated, but because we are paralyzed, we do not move those hands. Otherwise, we get in trouble. But sometimes when we have night terror, actually people end up beating their wives, or they get beaten by their wives because of the night terrors. So the visualization also can improve performance. And in our children's hospital, we are using this transcranial magnetic stimulation approach to enhance the performance of neurosurgeons. So through translation machines that, thoughts can translate, machine taps into motor program in a person or animal, imagines an act, and it decodes it into very distinctive electric commands to a device that puts the thought into an action. And you have seen, and we were talking about earlier about Elon Musk's, monkey playing those games. So those are actually these are the principles behind that technology that allows you to really take your thought process and convert it into distinctive, very distinct electrical command. And these commands could be then kind of relayed to computer devices to perform a particular action. So in our lab, what we do is that we have these robotic monkeys where we use these surface electrodes, and whatever Thomas is doing here, monkey will actually copy all of his facial expressions, his blinking, his actions. But what we can also do is that we can ask these students to really think about turning the light on or light off by looking at the screen on a different specific, letters or words, and they are converted into commands. We bring in young school children. We mount these electrodes on the scalp, and whatever this student will do, a child will do, they can even control a cube in a tunnel. So you can imagine that you can push the cube up or down, or move it down further deep into the tunnel. And this allows us to really have this interface whereby we could connect wirelessly with these devices. And for example, to control a car here. It's actually better than Google Car. It's only brain control, or to fly a drone inside the lab. We make sure that somebody's watching the door. The dean doesn't come into the lab and gets me in a lot of trouble that you're not supposed to do this. But we do this anyway. So on these drones, we built in these little sensors. These sensors are just like your GPS system, providing the actual electrodes, the precise detail and information about different objects that it is approaching or coming in contact. And so these sensors will continuously do this, and you can land these drones much more safely. When I gave this talk in Pakistan, I was told that this is no big deal. Our pilots are flying, aircrafts without licenses. So, it's not such a big deal. So really comes to how can we now take these technologies where we can interface with large networks of brain cells. We have the ability to be able to do wireless, communication between brain and a computer or an electronic device, or make a hybrid between these two. How can we take this into a clinic? So the first thing that really came to our mind is that non-invasive approach would be to really do this in a setting where things are already there. For example, in the case of epilepsy, we got together with a team of neurosurgeons and we asked the questions that when they plant these surface electrodes in the brains of those children at our children's hospital who do not respond to any medication, and then there is no way but surgery for them to really regain or to detect where seizures are coming from. These surface electrodes are mounted. There's a 30-foot cable. The child is attached to it. These kids will stay in the hospital for 10 days. And whenever they will seize, these electrodes will help determine as to where the seizures are coming from. And then surgeon makes a pretty educated guess to go in and take those foci out where the seizures are generated. The problem is that these electrodes are really not very sensitive, and oftentimes guesswork is needed whereby a surgeon ends up taking even the normal chunk of tissue out just in case they missed that particular area. And we also know that in 40% of the cases, even after surgery, these seizures will return because the circuit itself is prone to seizures. So what do? We actually teamed up to develop these electrodes, which are 300 times more sensitive than conventional electrodes. And we made them really very specific and plant these electrodes inside and then have them stuck outside whereby there is no really wires attached. It's completely wireless. It is also MR compatible. So once the electrodes are planted, the child can be taken into an MRI. An MRI image is created with a whole brain tomogram showing the positions of the planted flap of electrodes. And then whenever this child will seize, a wireless signal is sent to a portable device, which is a backpack mounted on the back of the child, just in a pocket or somewhere. And whenever this child will seize, not only it will pick up that seizure, with exquisite foci and really the specific areas, but it can also send a 3D tomogram to surgeon, telling the surgeon that this is where the seizures are coming from. And out of 30 seizures, 28 of them have really come out of that region. So we have done these experiments in animal models, validated. We prototyped it. We actually represented Canada in the falling wall competition in Berlin. We won the prize there, and the technology is really now ready for implanting. So I talked about a lot about, you know, the work that we have been doing and gave you one little glimpse in terms of where the technology is really heading. But these are really the people behind who have been involved in this specific project. Pierre, Colin, and we had Ryden, who was really the youngest pilot to fly. He's the one who provided us tremendous really insight into as to what pilot really sees in terms of flying the drone. He won the Government Journal's best gold medal as well. And many really brilliant students in the lab who have done PhDs with me. And then we have postdocs, and of course the boys will be boys. You know, they all kind of represent my lab in a sense. And they are all really done really phenomenally well. And they're really, really the brilliant group of people who have really contributed to this work. So for the last really few minutes, I would really like to talk about, I will skip this as to where this is going, but maybe I should.
In the near future, tragedy may come with new choices. Imagine you've been in a car crash that killed your daughter.
Is she okay? Is she okay?
Or has it?
Is she breathing? Is she breathing?
Hey.
Is she okay?
Time of death, 9:17. Your daughter's brain was not impacted by the accident. If you wish to scan her brain for digitization and upload, you have approximately 5 minutes.
Do it.
Now imagine the power you wield when you can order a digital copy of your daughter's brain. And do you know what comes next?
Hi, Dad.
One day delivered to your doorstep is an artificially intelligent android that will act, look, emote, just like your daughter. Every memory from birth on will remain intact, but she'll also be a walking computer with access to all the world's knowledge. This is what your world will look like. This is the future of artificial intelligence. It's the deep future. Your body, gone. You're all computer, all the time.
Hello there. May I ask your name?
Hi there, my name is Ignacio.
It's nice to meet you, Ignacio. So, what country are you from?
I'm from Canada.
It's pretty cold there, eh?
Meet Erika the android. She or it, depending on who you ask, is one of the most sophisticated humanoids ever designed. And she's giving us a glimpse of what a year-million AI might look like.
Modern computers are adding machine. They add very fast, giving the illusion that they're thinking.
So really the idea of showing all of this was that this is where the world is heading. This is where we are also thinking. Elon Musk, Neuralink, many other people are really thinking about how we can have artificial intelligence, machine learning, and all of these aspects that can then control robots and then your future medical doctors as well. So I always say that शहर को छोड़ के गांव की तरफ आते हैं, लाल जो है अपने वो वापस अपने वतनों की तरफ आते हैं, शहर को छोड़ के गांव की तरफ आते हैं, लाल थक जाएं तो माओं की तरफ आते हैं. So at the end we thought that how can we actually take this particular approach and then start something really remarkable in Pakistan and bring these technologies. So this really came with the idea to create a one-stop shop, which we call Knowledge Mall. And the idea is that if we could set up a multidisciplinary workplace for scientists, educators, entrepreneurs, industrial partners, and the commercialization aspects, and making it a one-stop shop, whereby all these different disciplines could really come together the way they were together in the Renaissance period and even in the golden age of Islam. So this is really what the vision is, that the central domain, the dome, will be what we call a knowledge mall, and partner with all the industry partners all across. So we have really thought of this vision as a Pakistan International University. And the idea of this particular university is to bring in all interdisciplinary areas under one roof. So it will have 5 different wings, and these wings could be artificial intelligence, machine learning, robotics, biomedical engineering, life sciences, chemistry, and physics. All these wings are really linked with the central dome, which is a knowledge mall. It's an open integrated learning system. And just like it happens in my own lab here, that we can actually look at the molecular aspects to bring students from engineering faculty who are working with our surgeons or medical doctors, and also basic scientists, to pull all of this together under one roof. But it should also be partnered with the industry, to have industry on site, so the technology that is developed locally at a particular place could immediately be taken to the industry, and prototyping done, and all the other aspects of commercialization done, from product development to releasing into the market. To realize this dream, we have purchased a over 60-acre land in Islamabad, and it's on the Murray Expressway just before the toll road. It is really beautiful area. It's a beautiful terrain, challenging at certain places, but it is absolutely gorgeous in terms of its landscape, its location, and its environmental impact as well. And the idea is to really build this particular entire complex in university, which will be interdisciplinary. We some constructions have already started. I was there before the lockdown, and we inaugurated this particular the wing, as the starting up the wing, met with many government folks to be able to really gauge their support and iron out many of these things. And now currently we are really fortunate that Prime Minister has assigned a task force that will really help us create a zone which will be research and innovation zone at one spot. So the idea is to make a one-stop or a knowledge mall in Pakistan, where we could have all these technologies, not only really train the future generation of highly qualified individuals, but also to be able to really produce our own products. And you can imagine that one BlackBerry can write off a city or a university. And I think this really what we envisage as a future. Pakistan has 50% of its population is under 25, very talented young people who are really dedicated to do some really cool research. But instead of sending them outside and not having a hub to land onto, our idea is that we should really get people to come to Pakistan rather than leaving Pakistan to learn various different things. I think I'm running out of time. We'll be happy to talk about what the vision and mission is, what the underlying objective and deliverables are, but I would like to leave that for any question and answer session. I really thank you for your wonderful hospitality and the opportunity to speak to you. I'll pass it on to Dr. Danish Bhatti or whoever is.
Thank you, Dr. Syed. Yeah, that's great. It was a wonderful presentation, very exciting, very energizing. इस तरह की presentations सुनने के बाद दिल कर रहा होता है कि कल ये काम शुरू हो जाए और सब कुछ जो है ना fall in place हो जाए. But so we cannot wait for this to materialize and start happening in Pakistan. Before I go on to my co-panelists and other questions, which we have a lot of, let me first invite our main panelists for the day. We have 2 panelists for today's talk. So let me first start with Professor Rashid Ahmed, who is the Associate Dean of Academic Affairs at School of Public Health in University of Illinois in Chicago. Professor Rashid Ahmed, your comments and any other introduction you would like to add to it, please go ahead.
Assalamu alaikum. I will be just very brief. It's exciting time for Pakistan, and I think the work what Dr. Naweed Syed is doing, I think is going to bring a lot of new innovation, and also the university what he is building there. I think we all need support in a way that we can all bring all the professionals together and also create awareness. So I will leave it here right now just to let other people asking the question. I'm happy to also respond some of the questions, but I will leave other panelists to discuss more.
Thank you. Let me invite my second panelist for the day, Professor Mustafa Saad Siddiqui, who is Professor of Neurology and Neurosurgery and Division Chief of Movement Disorders and co-director of the Deep Brain Stimulation Program at Wake Forest School of Medicine, North Carolina. Professor Siddiqui also chairs the Merit Research Subcommittee. Please go ahead, Mustafa.
Assalamu alaikum. Dr. Syed, that was such a fascinating talk, and congratulations for all the work. You really gave us a story, an inspiring story of translation of research from bench to bedside, and I must really congratulate you on this concept of knowledge mall, and you are practically now arriving in Islamabad with all those ideas. So really congratulations for that. So I look at brain, and this is in some philosophical thoughts about it. So I look at brain as the center of universe. I mean, there are so many similarities. We have 100 billion neurons in our brain. The world's population right now is about 8 billion, so not even close. Then there's another concept of 6 degrees of separation. So they say that every human being in this world is connected through 6 degrees. I mean, everyone is connected to everyone because I know someone, and then they know someone, and then they know the person. And the same concept happens in brain, where there's 6 degrees of separation, and it's all about connections, how these neurons make these connections. And unless there is a 15-year-old attending this presentation, everyone in the 90 participants I see and the panelists, unfortunately, I have to break this news to all of you that we have all lost a couple of brain neurons today. So we are all moving that, but how can I still claim that I can beat a young medical student? And that's all through connections we make through our experiences. So it's all about that. So think about this from a standpoint of when you think about neuroscience and the areas you want to research. The concept of brain-machine interface, I mean, it began in 1800s, and then Jasper and Penfield were the people who really published the first papers in which they found that when they were stimulating the temporal lobes, people were recalling the memories which they could not recall. Very fascinating read. Anyone interested, they should Google and read those papers. But really, for me, so I'm a neurologist, and I've been involved in stimulating brain, no pun intended, for the last 16 years. And for me, it was the clinical use of brain-machine interface, and that we saw in the form of deep brain stimulation, and it has advanced through the treatment for Parkinson's disease, tremors, and Alzheimer's now being under trial. So it's a fascinating journey, and Dr. Syed mentioned about the challenges about the closed loop and two-way circuits, and now we are recording brain activity, and through the brain activity, the behavior of the stimulating device gets changed. So it has been a fascinating journey, and all those 90 participants who are here, this talk which Dr. Syed gave is a great talk to motivate you into thinking about a career in neuroscience. So one simple question for Dr. Syed is, so I find the new field of optogenetics very fascinating with so much potential. Now that we are clinically actually modifying single neuronal activity at a 0.4 millimeter or less level, or actually at the axonal and the cell body level, when do you feel that the field of optogenetics, we would be seeing a clinical use out of that?
I think a beautiful description, really. And also, I think talking about the closed loop or open loop system, we also developed a specific electrode that could, excuse me, detect a single dopamine, from a synaptic terminal at the neuronal level. So the idea was that can we really connect now these electrodes, the DBS electrodes, with our sensors, that when certain amount of dopamine is released, that all of a sudden it will shut off the electrodes, bypassing the actual patient, stimulating the electrode themselves. So I think you're absolutely right that, not only the ability to record, but also be ability to release, different chemicals or drugs at the local foci to be able to manage, these diseases. So I think the fields are really expanding very, very nicely. And for optogenetics, we still, same holds true for transcranial magnetic stimulation. The biggest dilemma that, I lose my sleep is that I may stop a child from having hyperactivity or, ADHD-like symptoms, but I don't know if this stimulation is altering brain circuits somewhere else, at the expense of really the local stimulation. So same thing is with optogenetics as well. We don't know that if we are perturbing the natural patterns of connectivity and activity between brain cells, what could be the collateral damage that may come up in the years to come. And as you said that, we are losing brain cells, but in animal models, we see in the hippocampus, there are new neurons born all the time. And not only that these neurons are born, but they are preferentially recruited in the learning and memory circuits as well. And for humans, that is not really conclusive yet, whether we do produce these neurons in our hippocampus that are then used for learning and memory. But as you pointed out, that the synaptic connectivity is, plasticity is open. So we don't know if we're using optogenetics approach to express specific channel rhodopsin, whether it will change the intrinsic neuronal properties, the ion channel properties, or synaptic properties of those specific neurons. And as a consequence, we will change up the network properties as well. So I think in the animal models, you can alleviate pain, you can really make them pain insensitive. We also know that many people in Pakistan, they can stick needles through their mouth, through face, and they don't feel any pain because they have mutations in sodium channels. They don't really feel the pain. We can actually do this in rats and rats and monkeys, but I don't think ethically those experiments will be allowed in humans yet. So we are quite some ways, but it's a great, really great question, whether it could become an aspect of translation for obsessive compulsive behaviors, for people who really have serious psychological and neurotic behaviors, whether this approach could be used in those cases.
Thank you.
So, Dr. Syed, let me ask a few technical questions, maybe just a short directed one before we go into larger, bigger discussions. There are some questions related to your project about the knowledge mall that we will probably can take towards the end to conclude the session for today. And then I see at least one person have raised their hand. If any of the panelists want to make any comments or have a question, please raise your hand. And I will try to invite also people who are raising their hand from the audience. So a few quick questions, short, directed about the technical aspects of things. So one question was about, you mentioned in your diagram capacitors versus transistors. So the question was, how they interact with the neuron. Capacitor can store current and they probably just discharge and activate the neuron, but then how are we picking up the signal from the neuron, in what way, shape, or form?
So that's really a great question. So, normally when we have these multi-area electrodes. These electrodes are usually transistors. And they, whenever they will elicit an electric impulse, it will cause actually damage to the cell. It electroporates them. It punctures big holes in them. And this is why we moved away from that particular approach, because it was hurting individual neurons. What the capacitor does is the capacitor actually creates a positive charge. So you can imagine, I showed you the diagram of the synaptic terminal. When the impulse comes, it depolarizes the actual nerve ending, which causes influx of calcium into the presynaptic terminal. The neuron is depolarized and it releases its neurotransmitter. So what capacitor actually does is that it only creates a positive charge, assuming as if a neuron is being activated by another neuron at the synaptic terminal. So no current really passes because they are silicon dioxide, they're inert. So no current passes from transistor onto the neurons. So it doesn't damage or does anything. It only depolarizes it by creating a positive charge underneath. So when this neuron now fires, it will be depolarized. And if it depolarizes, it triggers spikes or impulses. They travel across the axon at the nerve terminal of its interconnected neuron. And when that neuron fires, we really have transistor underneath that can pick up that signal. So it's really a loop system whereby a semiconductor or a chip can talk to brain cells and they can talk back to brain cells. So the idea really there is that there is no current. It is just like non-invasively stimulating a particular neuron. And so when you look at hippocampus, actually hippocampus has a shape where we have CA3 to CA1 and CA2 neurons interconnected. We know where presynaptic neurons are. We know where the postsynaptic neurons are. Where the presynaptic cells are, we put the capacitors, and where the postsynaptic cells are, we put transistors. So whenever we will depolarize these neurons, it will be as if you have an intracellular electrode that is depolarizing the presynaptic neurons to activate postsynaptic cells. And because you have transistor at that end, they're acting as a second set of electrodes, which is picking up the signal.
Thank you. And one question about the scale of these things that we are interacting with. What's the size or mass of a single neuron? How much charge are we talking about? Voltage change. And then with the transistor is picking up the voltage alteration as a signal that an electron as neuron has fired as a reception of the signal. And does that induces any electrical change in the transistor? So those were a few questions lumped together.
So great question. So I think in this particular case, our electrodes about 7 microns, individual. So one neuron, for example, in cortex will be 12, 13, 15 micron, and the same will be in the hippocampus. So one particular neuron can at any given time sit on one of our electrodes. But the beautiful thing is that if you have 2 neurons that concurrently come in contact with the same electrodes, half and half, it will pick up only the largest signal from one particular cell, and it will ignore the other cells. So you're not really recording field potentials, rather recording from individual neurons. And so it is a capacitive change or a transistor. When it fires, it actually at the contact site, you have sodium channel clusters. When the ions flux in, the electrodes can actually pick up the technique. I didn't talk about our patch clamp chip, which was unique actually in the world, where we allowed these neurons to be directly interfaced with our chip, whereby instead of using a patch electrode coming from the top, we build it underneath, we fabricated it underneath. The neuron will sit on top and it becomes in isopotential with that particular neuron. So we could monitor ion channel activities of a single ion channel at a single neuron level. And these are really the electrical charges. That's what they're discharged or recorded through the electrodes.
And then a couple of questions, again, I'm going to link them together. Question are related to use of the electrodes to actually stimulate those neurons. Can we create signals into the brain, such as smell, touch, hear, vision, by giving in this instruction to the brain, and then can we take these, like you are culturing these neuron on chip, can you culture enough neuron to create a sort of a rudimentary computer, a neuronal computer by these cultured petri dishes?
So great question. These are $1 million question. I actually didn't want to scare people, but when you saw that template on our electrode, the pilot student who I mentioned, he won the gold medal, Governor General's gold medal. He said that the flight simulator, when the aircraft is airborne 30,000 feet, the pilots don't touch the aircraft. It is on autopilot mode. And there is a flight simulator that will fly this aircraft from Calgary and land it in Ottawa or even in London. So the flight simulator pattern, he recapitulated on the chip using over 100,000 electrodes. And he programmed it in such a way, and we were actually, before the lockdown, we're planning to do these experiments in collaboration with Air Canada to use our flight pattern made from neurons that are constructed from rat brain cells to be able to really simulate the flight pattern. The first thing actually I made them do was the Nusrat Fateh Ali's Qawwali. I said, well, use capacitor and transistors as a beat drum or guitar or tabla or clap. And then for the duration and the frequency, you can program them. And these neurons were dissociated, and you wouldn't believe that within 7 days, those neurons were singing that Qawwali, even though it's a tough thing to sing. But the neurons could be really reprogrammed because they are novel. And not only that you need actually hippocampal neurons, but you can also use cortical neurons to simulate these kinds of patterns that you can program. So absolutely, you could really design any particular behavioral output by stimulating these neurons in certain ways and in certain frequencies, and the duration of stimulation as well.
Let me ask a couple of questions again linked together about more of the role of these kind of techniques in the pathologies, not normal brain functioning. And then I will take a couple of questions from the audience who have raised their hand. So the questions are in terms of role in pathology. So, you mentioned very early on in passing about the brain map of neuronal networks. You alluded to the role of mirror neurons, where they activate when you're thinking of doing something rather than actually doing something, like reaching out for a glass. So the question is that how good are we right now in terms of maps of these regions of mirror neurons and placing these chips in treating pathologies, whether it be a role in strengthening memory in Alzheimer's, or role of treatment in movement disorders such as dystonias, or more of a neuronal level regulation of seizures rather than use of field potentials?
So I think, again, $1 million question. For many of these kinds of, you heard the story about this lady who has severe ataxia and she couldn't even sit or stand, she would just fall down. And so what they actually did was they planted 70 electrodes around on her tongue, and they coupled them with the helmet. And whenever her helmet tilted, it would shock her tongue. So when the tongue was shocked, actually, within 20 minutes, she could stand up and even dance. And after 2 months of really training, they completely switched her tongue into a sensory motor cortex. So the tongue actually took over many aspects of her sensory motor cortex. And then she was seen actually riding her bike. So the plasticity of the adaptability of your brain is really the key to be able to reprogram different aspects. Think about your sympathetic and parasympathetic nervous system. Our guts have more neurons than our brain, as Dr. Siddiqui pointed out, that tens of billions of brain cells. We actually have larger numbers, and those brain cells are like civil servants. They don't do much; they just sit there. But can they be really put to work some ways? And I think tapping into those neurons to be able to do certain behavior or behavioral control is absolutely feasible, and it can be done. The problem really is that, for example, children, when they come to our children's hospital, and these children are having, either panic attacks or they have obsessive compulsive repetitive behaviors. You put this transcranial magnetic stimulation non-invasively. You fire off the magnetic pulses, and these kids can just sit and calm down without actually doing anything. But I always worry about what are the alternative changes that we're creating in the brain in different other parts that are not really have anything to do with a particular behavior, but it may make them, maybe a serial killer, or make them something really, really bad. And as you know, that, Dr. Siddiqui can tell you better than I would, and yourself as well, that even the surgeons are being trained using the transcranial magnetic stimulation to improve their surgical skills in the OR. So I think, many of these technologies, they're okay as long as they're subcortical. But, going deeper into the brain is really, that's what worries me. And this is what is, we have very stringent rules and regulations in that case to be able to really break through in terms of FDA approval. It took us very long time to be able to do this. They wouldn't allow us to test our chips. So we partner with our neurosurgeons who are planting surface electrodes. And we ask them, could you at the same time plant our electrode? And then we will really decipher their efficacy. So I think, at this moment, if we turn our electrode for epileptic seizure detection from a detection, a diagnostic tool, to be able to interfere and become a really instrumental. So give you an idea, the moment an electrode detects seizure, it will discharge, but at the same time, it can activate a capacitor, which is located adjacent to it. Capacitor can now send the negative hyperpolarizing current pulse to really offset the spread of seizures. We are in negotiation with Apple for iPhone maybe 16 or 18, where a sensitive cochlear implant could be planted. And this cochlear implant, because it's very close to temporal, very close to a really brain region, when it picks up the signal, it dials your cell phone and it tells you, Naweed, you're about to have seizure, sit down. I just dialed 911. And on your phone, I have turned on the GPS locator. The ambulance exactly knows where you are and the help is on your way. So I think these are still non-invasive approaches, but anything that we go more invasive, a lot more work is really needed.
Thank you.
And Danish, can I just add a few comments on the same thing?
Yeah, very quickly, Dr. Siddiqui, I have tons of questions and we're so over time.
Yeah,
Please.
so Dr. Syed, he exactly, his concern about what happens in upstream and downstream, where you are making it an interface and the behavior for that. And my just comment is that we have now the technology to stimulate or change the neuronal activity at a single neuron level. The question is, we think that we know a lot about brain, but we know very little. And it's knowing about the circuitry of different diseases. That's where the answer lies. And that's our next frontier.
Absolutely. I think, really, maybe I left part of the question unanswered about the Alzheimer's disease and learning and memory, whether learning and memory can also be transplanted. So our first chip, really, we recapitulated learning and memory on our chip. And that was really, I couldn't sleep for several days when I actually saw that we could train a neuronal network. And that memory that was between two neurons, the synaptic plasticity was captured by our semiconductor chip, and it retained that memory throughout. So if you can really program these networks to exhibit, or this neuroprosthesis or artificial chips to exhibit learning and memory, one would then might be able to recapitulate learning and memory in instances for Alzheimer's disease, for example.
Thank you. Let me invite Dr. Tariq Zaman for his question.
Thank you, Dr. Syed, for a wonderful talk. I'm a big fan of your brain and computer interface, especially the idea of knowledge mall. So it has innovative application towards the brain manipulations. I would love to be the part of your team because I would like to serve Pakistan. So currently I'm a neuroscientist at Michigan State University. I have 16 years in neuroscience research. So most of my research interests like epilepsy and autism and expertise are very much like you. For example, gene targeting, cellular to network level physiology, synaptic plasticity, in utero gene manipulations in developing brain, and to manipulate the developed one to rewire the brain. But my bigger focus is the precise and personalized medicine. But your approach is quite interesting, and there is no one that who can even close to you. So that's why I always follow you. So I just want to share one thing, because my son was very much interested in your approaches. He actually got the idea from your research, and he got the gold medal at the school level, means that the board level, basically. So he presented in a similar way. But anyhow, I am your big fan, and I'm very much into research. So my question is very much simple, and I'm actually very much curious about the biocompatible electrodes, because I was working once in Korea before Canada and US, basically. So there was one group was developing graphene-based electrodes that can sense better the current. And just one thing, I'm actually curious whether you have grown the iPSCs, that's induced pluripotent stem cells, on chip, then implanted in the brain that might overcome the mechanical damage to neurons. Thanks.
So great question. Actually, I'm also, I follow your work, and mashallah, you're doing really well. And I think it's a tremendous joy to see our younger generation really working at the cutting edge and really doing phenomenal research. So the question is that, can we really interface these technologies for stem cell induced neurons? Absolutely. And I think that really is the next step, is that we could perhaps use maybe transgenic approaches to produce these cells that could be from humans, and then try to see if we could really monitor their activities of these neurons in a really natural sense. But in terms of going to this plan for Pakistan, we need really all of you. This is not my individual project. This is really a national pride. And we wanted to really bring back the golden age of Islam, whereby we have all scientists who are all polymath working together to solve real world problems. So I think our idea will be, for this particular initiative, is that we will bring all the top-notch scientists and scholars to be the directors of different programs. There will be no local high-level appointments, but all the directors will be overseas Pakistanis who have expertise in that domain. We will fly them in every 3 weeks or a month. They will come and they will check the progress. If it is not satisfactory, the local folks are fired. We are also getting internationally recognized educators to come and teach a particular course. And for the first 2-3 years, we will have local teachers actually sit in the class and learn from the expert as to how teaching is really done. So at all different levels, the idea is to really bring back the golden age of Islam, where we were performing cataract surgery 600 years before Europe, and we knew human anatomy 1,000 years before Europe. I mean, where in the world you see polymaths coming that the Islamic world had really produced. And the reason there was the integrated learning, where everybody was working together to solve real world problems. So we really welcome you. We can really discuss all the nitty-gritties offline. And I am in contact with you too, and may Allah bless you and keep doing really what you're doing. And I think also encouraging our younger generation to really embark on this field, which is uncharted territory. It scares them. Most of the time when we studied, most people studied their medical school, they were scared of brain. But I think now we can make it more interesting and exciting for them.
Let me invite Dr. Zafar Hayat for his question and comment. Please go ahead and unmute yourself. Dr. Zafar Hayat, please go ahead.
Thank you very much. And are you hearing me?
Yes.
Are you hearing me?
Yes. Yes.
Hello? Okay. Thank you very much. Very comprehensive and technical, rather I would say ultra technical neurological in-depth research on the brain, which is a very versatile organ. You know, other organs are having single function, like heart has got pumping, the lungs have got ventilation and oxygenation, the GI have got digestion and absorption, assimilation, like that. But the brain is a very versatile organ in that it controls the whole body. All the body centers are located in the brain. So to be very brief, I'm basically a physician with internal medicine and especially interested in neurology for a long time. So I would say that be aside of the technical know-how, which we went through, and the optogenetics and everything, how we control the neurons are to stimulate or inhibit them to achieve our functions of the brain or retain the function of the brain which they were lost. I would be really amazing for me that apart from the technical know-how, I would come to the functional or the clinical uses in the patient phenotypes. Like what would be role in, I would say, a lot in depression, manic depressive psychosis, epilepsy, many other disorders of the brain, like autistic child, autism, etc. But I would be really wondering whether it is of any good in organic brain diseases, like I would say degenerative brain diseases like the motor neuron disease, Parkinson's disease, multiple sclerosis, and these sort of disabling diseases, or the nerve deafness, congenital nerve deafness, whether you would be able to stimulate directly the temporal lobe for hearing, and also from beginning, like in the development of the child, after having identified that they got congenital deafness in infancy, would we be able to, treat such sort of patients from the very infancy, like the congenital deafness, through to.
Thank you, Dr. Hayat. Dr. Syed, if you want to quickly comment on role in pathological conditions.
I think really a great question, very in-depth, very comprehensive. But the thing is that, if you have a bulb or a switch that were connected, if the bulb is fused, you can change the bulb. And if the switch is broken, you can fix the switch. But if there is no wiring, there is no real point in terms of really turning on, changing the bulb or changing the switch, because wiring is not there. For many of these congenital or developmental disorders, when the wiring is not there, I don't think that any of these technologies will be able to help them at this stage. But this is at this stage. But you move it forward, whereby you could control, wirelessly many of these patterns of activities in the brain. I think sky is really the limit. We have no idea in terms of learning and memory dependent mechanisms in the brain. We don't know about the conscious and unconscious part of the brain. As you know, that the conscious part of brain only scans 42 frames per second. We can deal with this, but the unconscious part of the brain does 1.2 million frames per second. And those are based on individualized personal experiences and lifetime activities. And that is where the precision or personalized health is really coming in play, because this becomes very, very difficult to decipher as to what particular thought in an individual's brain is coupled with the past and in the future, and then how brain is integrating this information to be able to develop really precision health or personalized devices for those. We are not there yet. It will take really very long time for us to really even understand unconscious part of the brain and then the subconscious. To give you an example, if I ask you, where's your foot? Up until now, you didn't know where your foot was, and now you started to wiggle. And so that was really the major challenge of the unconscious part of the brain, which controls so many different functions, and yet they are not recognized by our really awareness, our consciousness at that particular time.
And lastly, let me ask you a couple of questions grouped together about your project, the Knowledge Mall in Pakistan. People are curious about what stage of construction it is, what's the timeline on it, what kind of government oversight contract or support or MOUs you have, how can people track the progress, where can they remain updated, and what other open resources are available right now for someone to get started in this field?
So great questions. I mean, I chaired a meeting of the Business Chamber of Commerce when I was there before the lockdown to actually bring industry on board and invoke their interest, whether they will be interested in setting up a shop on our facility. But because it becomes a commercial zone, we had to really reach the government, and Prime Minister Imran Khan has been really updated, and he has assigned a task force that's working with us to create a zone that will be innovation, couple education, research, and industry zone. So we have really started some construction already. There are some phases of the building are going up. The site planning is done. We had to realize that there is very shortage of water in Islamabad. So we had to really make sure that we have provision of our own lake. Well, the lake will be generated. It will be all solar panel driven. It will be the largest library, digital library in the country. We will have the largest auditorium in the country as well. And so when the actual road construction started, alhamdulillah, there were 7 fountains of huge reservoirs of water erupted. So we knew that, there is a force behind all of this that actually wants this to happen for Pakistan. So we are thinking about really, the bulldozers are right now on the ground, the flattening, because it's a very difficult terrain. We had to really also think about the earthquake zone. We have to think about the animals that we will really have. The animals, as you saw in the picture, are already there. We're bringing, these very exotic animals from different countries to make sure that, we have cows that can we can also enhance their capacity, local buffaloes to be able to produce more milk. Similarly, sheep we brought from New Zealand and Australia. So we will dig into the mountain areas to build the animal housing where the animal research will be conducted on site. So the timelines we're thinking about before the end of this month, we will be able to really get the main part of the building, the first floor is done. We are asking government to give us 20 years tax-free exemptions. We're asking government to provide us with no import duty. We are asking the government to allow a state bank to release funds without actually questions answered, because right now the oversight on that is really scaring major donors. And once we really have all of this major infrastructure on the ground, we will have to do the fundraising. And I will invite all of you, and there's an opportunity for naming right as well for the university, for different units, different floors, so people who contribute. This will be actually people's project, and this will be done independently from government. And we also ask the government, the local provincial government, to support us in terms of electricity, water, and gas charges as well. So, many of these things we would have been really in a much better position had this not been because of the COVID-19. So the work in progress, and you will, inshallah, soon hear some very good news.
Thank you so much. So I think we can conclude the session here. I typically invite Dr. Syed Uzair to do the conclusion and give concluding remarks. But I believe, Dr. Uzair, if I'm not mistaken, we also have presence of Mr. Abdul Hamid, the Consul General for Pakistan in Toronto. And if he's still present, and if I'm not mistaken, maybe we can invite him to do the honor and give a concluding remark for the session today. Mr. Hamid Saab, please go ahead.
Thank you very much. In fact, I was supposed to be a speaker. I was an attendee because I'm a fan of this Dr. Naweed Syed's amazing project of establishing university in Pakistan. It in fact inspires so many people around the world, because the idea of having a knowledge mall, a multidisciplinary place of exchanging ideas, right from artificial intelligence to medicine to industry, it's amazing. And his other ideas like Muslim Renaissance and integrated learning and of reviving the idea of polymath, it's amazing. And I must say that I have learned a lot. I have benefited a lot from today's conversation, and it was so inspiring. I in fact can share my notes, and I would surely convey to Government of Pakistan as well that how wonderful conversations are going in North America. From like some of neurons, as a civil servant, I might be sitting idle, but I would be conveying this much, that here we are having a wonderful conversation. So nice of you. Thank you very much.
Thank you so much. So we can now officially conclude the session for today. And thank you so much, Dr. Syed, for joining us. We have sent you a link for another meeting where we can hear how we can help and collaborate with you. So we can join there later on. Thank you so much, everyone. Thanks.
Thank you. Thank you for your